In this lecture, we're going to talk about the cell cycle. The cell cycle has got two phases. There is an interphase and there is a mitotic phase. During interphase, the cells prepare for division, and during the mitotic phase, the cell divides into two daughter cells. Now let's look at the first phase, which is an interphase. This is kind of the preparatory phase, and it consists of three stages: G1, S, and G2. G1 is the first growth phase. During this phase, the cell growth happens. Synthesis, or S phase, is the second one, and during this phase, the cell duplicates the DNA. G2 is the third part, and this is you can consider this as the second growth phase, and further cell growth happens, and the cell is getting ready for division. During this phase, all the DNA replication errors are corrected so that it is not passed on to the daughter cells. Once a cell is ready to divide, it enters into the next phase, which is the mitotic phase. Mitosis has got four stages: prophase, metaphase, anaphase, and telophase. Telophase is followed by cytokinesis, and it is during cytokinesis that the cell physically divides into two daughter cells. Now the question is, why do cells undergo mitosis? Cells undergo mitosis for a variety of reasons: for growth and development of the organism, to replace cells that die, and also to repair damaged cells. Now let's look at each stages of mitosis. The first is the prophase. So this is the prophase, and you can see during prophase, the nuclear envelope starts to disintegrate, and the genetic material is present as chromatin threads. These chromatin threads starts condensing. So now they are condensing into compact chromosomes. During the next phase, which is the metaphase, the chromosomes align along the equator of the cell, and then the centrioles, they start migrating to the opposite poles of the cell, and the centrioles sprout long microtubules, and these tubules are called spindle fibers. Spindle fibers attach to the centromere of each chromosome. So you can see this is a spindle fiber coming from the centriole, and this spindle fiber is attached to the centromere of the chromosome. So the centromere is that part of the chromosome that holds the two sister chromatids together. The next phase of mitosis is called the anaphase. This is the phase when the two sister chromatids are now pulled apart. So the enzyme cleaves the two sister chromatids apart, and you have two daughter chromosomes. The daughter chromosome starts to migrate to the opposite poles of the cell. So that completes the anaphase, and then we are going into the telophase. During this phase, which is the telophase, the chromosomes reaches the opposite poles. So you can see the chromosomes have already reached the opposite poles of the cell, and here the chromosome starts to unwind and uncoil to form chromatin. So it goes to its previous shape, which is the chromatin, and they exist as fine threads, and at this stage the nuclear envelope also starts forming. So that's the telophase. The next stage of mitosis is called cytokinesis. Cytokinesis is the stage where the cells physically divide into two daughter cells. So there is a complete separation of the cytoplasm into two daughter cells. So here you can see the two daughter cells are now separate and they start functioning independently. So this completes the division of the cell. So as a recap, there are two major phases: the preparatory stage, which is the interphase, and then there is the next stage, which is the division phase, that is a mitotic phase.